Hi and welcome Mindsetters to Learn Extra. Now that you've learned how to deal with genetics and some history on genetics, let's now look at how we can solve some of these genetic problems. Remember, once you learn the basic pattern on how to present your answer, half your battle is won. What I'm going to show you with the progression of problems that we're going to do is that the pattern does not change. We may change the symbols that we use for the different types of problems, but the actual presentation of the problem is the same. You have to know the format because the format gives you 2 marks out of the possible 6 marks in a genetic problem. So make sure that you are aware of that format and you are conversant with it so that you can deal with it as you're going along. So we're dealing with genetic problems. What must you know? What must you know to do and what must you be able to do? You must be able to solve monohybrid's genetic problem. And this is the core of this section. The more monohybrid questions you go over, the better your understanding is going to be, and the better your understanding, the more marks you're going to get. Genetics is plus minus 45 marks in the paper. Determine the proportion and ratio of genotypes. Obviously, that's a follow-on onto this part here. Once you work out the genetic problem, then at the bottom, once you have your answer, you can then have the ratio of the genotype and phenotype. Sometimes they ask for this in just simple ratio in whole numbers, and sometimes they ask it in percentages, so you must know both. By using examples, you must be able to distinguish between complete dominance, and that is what this lesson is all about. We're going to do a problem in almost each of these so that you can see what's happening. Here, one allele is dominant over the other. So the other obviously is recessive. So you get a dominant allele and you have a recessive one. When we put these 2 alleles together, remember each allele codes for a particular characteristic in a particular way. So if we put a dominant one with a recessive one, then what we would be able to see on the outward, that means the expression of these alleles, would be the dominant one only. I use the simple phrase, when the cat is away, the mice will play. Meaning that as long as the dominant one is there, you won't be able to see the recessive one. We say that the dominant allele masks or shadows the recessive one. So we must know that. Incomplete dominance means none of the 2 alleles are dominant over each other. None of them. And in the result, we would see a new blend forming. And codominance, both alleles are equally dominant. Co-partners, that means both the partners are equal in the business. So here codominance, they are equally dominant. So those are the 3 things that you need to know there. Further, you must also solve genetic problems where these 3 types of dominance are shown. Which 3 types? These 3 types here. You must be able to differentiate between sex chromosomes and autosomes. Sex chromosomes are those that determine sex. That means the gender, maleness or femaleness. Autosomes are all other chromosomes besides the sex chromosomes. In humans, that will be chromosome set number 1 to 22 autosomes, and sex chromosomes is the pair labeled 23. Represent a genetic cross to show the inheritance of sex or inheritance of gender. What is meant by multiple alleles and how this is dealt with in blood groups. And we must be able to solve problems where the blood type is involved. We must also be able to, and again this differentiate sex chromosomes and autosomes comes in because it's now linked to this part here, what is meant by a sex-linked characteristic. We must be able to solve genetic problems involving sex-linked characteristics, especially hemophilia and color blindness. Remember, hemophilia and color blindness is what is prescribed in the CAPS document. So in other words, for these two, you need to know that this is sex-linked. If the examiner chooses to test you on another sex-linked disease or sex-linked disorder, they would have to tell you that it is sex-linked. But the way you work out the problem will still be the same. You must know what is meant by a pedigree diagram, and you must be able to deal with a pedigree diagram. In other words, you need to interpret a pedigree diagram. Further, you must be able to state Mendel's principle of independent assortment. Remember, in the first segment of genetics, you learned the law of dominance and you learned the law of segregation. Now, we are talking about another law called the law of independent assortment. Do not confuse this with a random assortment. A random assortment we talk about in meiosis. It's not the same as independent assortment. In independent assortment, we talk about the fact that if an allele is on a different chromosome, then when these separate, they do not affect each other. So when they assort, one characteristic is, or one allele is not affecting another one. You must be able to solve dihybrid genetic problems, and you must be able to determine the proportion, the same like when you deal with monohybrid crossing. In the end, you must be able to say this is the phenotype-genotype ratio. In dihybrid, we are more interested in the phenotypic ratio. Let's go straight into exam type of questions to deal with those topics that we spoke about. The first one, question 1, justify the following statement. That means the statement is being made, the statement is true, you must justify it. You must be able to give us some proof to show that the statement is true. Sex chromosomes are largely responsible for the fact that in human populations, there are approximately as many men as there are women. They're not telling you must say whether it's true or false. They're telling you that the statement is true, you must justify it. It didn't say critical analysis of this question is required. It didn't say criticize it. It didn't say evaluate. It said justify it. So it's telling you that the statement is true, prove it. And they didn't say give a genetic diagram, they just said a Punnett diagram. Now remember, for sex determination, we look at XX and XY. XX, female. So in other words, in a human being, or let's talk about human beings only. In a human being, if chromosome pair number 23 is XX, that will be a female. If chromosome number 22, set number 23 is XY, that would be a male. So if we are working it out, obviously if fertilization has to take place, it can take place with a male and a female only, not with two females or not with two males. So if we have a female XX and a male XY, what are we going to do? We're going to say X and X gives us XX, X and X gives us another XX there, X and Y, XY, XY. Notice 50% chance of a female and 50% chance of a male. So that is justifying that answer there, and to justify it we must conclude there it's yes, you're getting 2:2, 2:2 XXXY, therefore 50% will be women and 50% are men. So that justifies that statement there. The next question, Question 1.2, still dealing with monohybrid crosses. In dogs, there is a hereditary deafness caused by a recessive gene D. So first of all, deafness is caused by a recessive gene. The fact that we use a small letter, that should already inform us that it's caused by a recessive gene. A kennel owner has a male dog that he wants to use for breeding purposes if possible. The dog can hear. So the owner knows his genotype is either DD, capital DD, or capital D small t. In other words, what are we saying here? We're saying that he's either homozygous or heterozygous. What does that mean? Homozygous means the two alleles are identical. They both capital Ds. Or they could be two small d's. It'd still be homozygous. But this is homozygous recessive and this one is homozygous dominant. Or the organism can be heterozygous. That means the alleles are not the same. They're not identical. They're coding for the same thing, deafness. But the one allele is coding for normal hearing and the small d is for deafness. So this individual is heterozygous for this condition. And using our law of the cat and the mice, the fact that it has the normal gene, which is dominant, that means this dog will be able to hear. Why? Because the dominant gene masks or shadows the recessive one. Now the problem is, if the dog's genotype is capital D small d, which is heterozygous, the owner does not want to use him for breeding purposes, because if this dog is mated with another heterozygous dog, then there's a 25% chance of them having offspring who are deaf. And this is a kennel owner. He wants to make money with dogs. Nobody's going to buy a deaf dog from him. Okay? This can be tested by breeding the dog to a deaf female. Use a genetic diagram to illustrate this cross. So we want to know whether this dog is heterozygous or homozygous. The simplest way is to cross that one with a homozygous recessive dog. So if the female, or rather let me just do that, the female is deaf. Phenotype, remember? Phenotype refers to the characteristic, the observable characteristic. Is the dog normal or is this dog deaf? The genotype, if this dog is capital D, small d, and the female is homozygous for deafness, then that is the situation. Meiosis, remember, you're getting 1 mark if that whole line is true, correct, and if that whole line is correct. Then you're going to write meiosis. Normally, we write meiosis here to show that when meiosis takes place, these alleles separate, and that will become D and small d, and these will be 2 small d's. All we're doing in this step of gametes is we're showing that in each gamete there can only be one of these 2 alleles. Why? Because when meiosis takes place, these alleles separate. And when fertilization takes place, that one and that one will give us D, small d, and that one will give us D, small d. And if we take the second one, small d, small d, small d, small d. So here now in this particular case, because they were both not heterozygous, it was heterozygous with homozygous recessive, what can happen? 50% will be normal and 50% will be deaf. So phenotype ratio, the phenotype is 2 normal and 2 deaf. If we do it on a Punnett square, we're going to have capital D, small d, small d, small d. This one here will become capital D, oopsie, okay, capital D, small d, and capital D, small d. And here it will be all small d, small d, small d, small d. I find it easier to work with the Punnett square because it's neater and it's less chance of you getting confused. So that's easiest way to do it. Obviously, all of this will change if this dog was normal homozygous. Then our Punnett square is going to show something slightly different. We will have capital D, small d, small d. Then all the offspring would be heterozygous, but they will all be normal. This is what this kennel owner wants to know. But obviously it doesn't mean that if all the offspring that came out of that dog were normal, then he's homozygous immediately. Remember, all the genetic Punnett square is doing is showing you the probability of what can happen. So it's important to know that we will have to repeat this investigation several times to check the reliability of our investigation. So that dealt with complete dominance, where one is dominant and the other one is recessive. We're using a very complicated way to show that. Remember, there are simpler examples that you can go over in the past papers to deal with complete dominance. Now we are looking at something different. They say a homozygous snapdragon plant with red flowers was cross-pollinated with a homozygous snapdragon plant with white. Notice immediately this is slightly different to what we've been seeing. They're showing a capital R for red and a capital W for white flowers. That means this is not complete dominance. But we have to see the results to determine whether it's incomplete or co. Let's carry on. All the plants that grew from this cross had pink flowers. Is it red? No. Is it white? No. It's pink. So this is a new blend. That means neither red nor white is dominant over each other. Therefore, this is incomplete dominance. So important to see the results. Represent a genetic, not a Punnett square, but a genetic cross to determine the genotype of the F1 generation of plants. So we had homozygous red with homozygous white. Now we need to look at it. Remember to make sure that your capital letter W looks like a capital W so that you can deal with that as it happens. So let's deal with the problem now. So what did we have? Phenotype was red and white. The genotype was RR, homozygous, and WW, homozygous. The gametes, meiosis is going to take place. The gametes are R and R. Here, even if you put one R, because there can only be R on this side, and W and W, capital. Then you will write fertilization. We didn't complete it in the last one, so we're going to do it here. 1 mark for that, 1 mark for that, 1 mark if the gametes are correct. Now, if you write fertilization and meiosis in the correct places, that means meiosis there or here, just to show that it's happening in this step, and fertilization with the gametes, then you will get another mark for that as well. And in this case, it's clear that because they are homozygous, there's only one possible combination. It's R, capital W, and they told you here that it was pink. So therefore, your answer there is 100% are pink. And obviously, you can do the same in a Punnett square. Capital R, capital W, all of them will be RW, and therefore you're going to show that. Here's that other marks that I'm talking about. 1 mark for putting P1, oh, I forgot about that one again, sorry. P1 and F1, that's another mark that you can get in its right place, and meiosis and fertilization. Therefore, I told you at the beginning that if you know the format, half your battle is won. Not half, but 1/3. It's 2 out of 6. Most genetic crosses are worth 6 marks if it's given in this format. And if you got the format right, even if your answer inside is incorrect, but if you got P1 and F1 where they belong, or P2 and F2 if it's a second generation, and meiosis and fertilization in the right places, you would get 2 out of 6. That is 33 1/3%. So that's better than nothing, obviously. So this is a sample question on incomplete dominance. Now we move off one step further. And this is a little bit complicated, so we need to be sharp when we're working here. What do they say? They say Mrs. Johnston and Mrs. Johnson. And, sorry, Miss Johnston and Miss Johnson and Miss Johnstone. Oops, look how close those surnames are. Johnston, Johnson, and Johnstone. They all entered the same hospital and gave birth to baby girls on the same day. All of them had girls. And all three babies were taken to the nursery to receive care. Someone later claimed that the hospital mixed up the babies. The wrong parents got the wrong babies. Ah, that's sad. Okay? As a hospital administrator, it is your job to make sure that each pair of parents has the correct baby. So, you order blood typing to be done on all parents and the babies. The results are as follows. Mrs. Johnston, blood group A. Mr. Johnston, blood group B. This is one set, one couple. The second couple, Mrs. Johnson and Mr. Johnson, O and B. And the third couple, Mr. Johnstone and Mrs. Johnstone are both blood group A. So what have they given us? They gave us the phenotype, the blood group type. We need to figure out the genotype. The babies, baby 1 is blood group O, baby 2 AB, and baby 3 is blood group B. Now, these are all the genotype, the phenotypes they've given us. Now remember that blood groups, the genotype A can be IA, homozygous, or IAI, heterozygous. B can be IB, and IBI. Notice we're using capital I and capital B for B and capital I and capital A for A. That tells you that A and B have some form of dominance. Blood group O, on the other hand, is given as two small i's. What does that tell you? That tells you that this is recessive. And then we get blood group AB, and that is IA, IB. So can you see the form of codominance shown here? That AB, when they put together, both show up in the individual. It's not a new blend. It's A and B. So this is known as codominance. So that means they are equally dominant, that's one. Notice also that normally we spoke about red or white, yellow or green, tall or short. There were only two allele varieties. Now here for blood group, we're getting blood group A, blood group B, blood group O, and different alleles. We're getting IA, we're getting IB, and we're getting I. There's more than two. This is an example of multiple alleles as well. So two things that are being multiple alleles. Two things that have been highlighted here. One is that there are more than two varieties of the alleles. So it's multiple alleles. But remember, in one individual, there can only be one, be two, sorry. Any two. It could be two of A, it could be A with O, it could be A and B, AB, it can be two Bs, or it could be B with O to give you that type of variety in that individual. But it's controlled by more than two, so therefore this is a form of multiple alleles. At the same time, we're seeing two classic examples here. When we see IA, I, or IB, I, what does that tell you? One is dominant, one is recessive. So this is a case of complete dominance in this particular example. When A and O or B and O get together, we see complete dominance because when it gets together, we say that the person is blood group A or blood group B. We don't say is AO or BO. The O is not showing. Why? Because A and B are dominant over O. And when A and B come together, IA, IB, we say that the person has AB. So therefore, they are both showing. This is an example of codominance. So in this one example, we see complete dominance with A and B over O, and we see codominance with A and B together because they are equally dominant. Okay? But now, let's go back to our problem. We're going to take one of them and we're going to develop it. First of all, baby that has blood group O can obviously go to any one of the parents because O is II. This one has A, that one has B. They could be heterozygous for A and B. Therefore, there is a possibility of having a baby with O. This one is B and that is O. This B could be heterozygous, so there is a possibility of having this baby. This one is A and A. They could be both heterozygous for A, so it's a possibility for O. So that's a difficult one to go through. So that's not an easy one to go through. However, the child that has AB can obviously not belong to Johnstones because neither one of them has an allele for B. Because if they are A, they can either be IA or I, homozygous for A or heterozygous for A. So the only alternatives that they can have children. Sorry, I'm showing the wrong thing in the wrong area there. Let me take the arrows away. Let me take all that whole work out. So let's go back down further, sorry. Here we're talking about these guys now first. They can either be IA or II. So what are the possible babies that can come out of that? If they're both homozygous, they can only have A, blood group A. If even one of them is only homozygous, they can still only have blood group A children. But if both are heterozygous, there's a 25% chance that they can have a baby that is O. So, like I told you, they can have any, blood group O can go to anybody. But these parents cannot have AB because there's no chance. Let's look at the next set of parents. Can they have AB? No, because they have Johnson is B and Johnson, the father is O. So they can either have children that are B or they can have children that are O, if this is heterozygous, if Miss Johnson is IBI. If she is IBIB, then all their children can only be B, because B is dominant over O. Because the Johnson here can only be small i's. So they can't have an AB child. So the AB child can only belong to these guys. Already logically you can see that. And blood group B, because AB can only happen to these guys, that means this child is already sorted out. So the child that is blood group B can't belong to this set of parents. So this child here can only belong to those parents there. Now show a cross in that. Now we can even show all of them there, or we can show one. So let's look at an easier one there. Let's take Mrs. Johnston, and we assume that Mrs. Johnson and let me go back to that. What's that? Johnstone, sorry. Johnstone. So we're looking at Johnstone now. They are both blood group A. So their genotype is IA. We assume that they're heterozygous, for example, IAI. So the gametes can only be IA and I, both sides. And when we put that here, we got IA, I. IAIA will give you IA, IA. IAI will give you IA, small I. And the same is going to happen the other way around. IAIA, I, but on that side, that one and that one gives you II. So they have a 25% chance of having a baby with blood group O. If we take Mr. and Ms. Johnson, it was, sorry, B and O. It's easy, O and B, and we assume that they are heterozygous. And there, obviously, they can't be. It can only be two small i's. So here we're getting the gametes IB, I, and here we're getting the gametes small I, small I. I put a semicolon to show that there's a separation. You just need to leave a space between them to show that separation. So IBI gives you IBI. And IBI will give you the same again. And that I and II, and II. So they have a chance of having a child with B and O. Okay? And the last set of parents, that was Mr. Johnston and Mrs. Johnston, A and B. So A and B. Here again I'm using, let's use homozygous here. Let's say they're homozygous. IA, IB, IB. Remember, they could be homozygous or heterozygous. You can show both. I'm just showing homozygous because we've looked at other heterozygous examples already. IA, IB, IB. And therefore, IA, IB, the answer will all be the same, IA, IB. So the AB child can only belong to these parents, and that is where you can solve the problem of that. Remember, this is a classic example of co-dominance. Why? Because A and B are equally dominant, and in the phenotype, both show. And with that, I think you've deserved a break. Take this break, refresh, and I'll catch you after the break. Hi, Mindsetters, and welcome back. I'm hoping you refreshed yourselves and you are ready to tackle the next part of our program. At the same time, I'm hoping you're not worried in terms of the blood groups and you're trying to figure out who's your parents, etc. Talking about that, there's an interesting case that's going on at the moment. We have parents where it has been proven that the babies have been swapped at birth and it's 10 years on. The one set of parents are saying, no, they don't want to let this child go. Interesting to see what happens in that court case because it will set a precedent in what's going to happen in the future. Again, what can be tested here? If we give you such a case study, we can test your opinions and ethics, etc. So think about that one. Our next question is dealing with pedigree diagrams and sex linked at the same time. So we're killing two birds with one stone. And the thing here is we're looking at pedigree diagrams and sex linked. Remember, when you're looking at sex linked, we must use the symbols XX and XY. Remember, in blood groups, we had to use the letter I, capital I or small I. Okay, now here we have to use XX or XY. XX if it's a female and XY if it's a male. Now let's look at this particular one. Males are squares and females are the circles. So what do we do in this one before we even start? We start putting there by the males X, Y and the females X, X. X, Y, X, Y, X, 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 Y. And so we go on with this one here. X, X, Mo, X, Y, and X, X there, and again X, Y there. Why we do this is to remind ourselves that when we are giving genotypes of sex-linked disorders, we must put XX. We can't just put capital H, small h. It doesn't cut it. Because here, the way this disease is transferred is determined that way. So they tell you, write down all the possible genotypes of the following individuals. Jubes, Sabi, Yusti. Now, here the thing tells you, a normal female, a male hemophiliac, and a normal male. So when we find the hemophiliacs or the males, it's easy. The males are easy to do. Because if the male is hemophiliac, we have to add H, small h, and if he's normal, we add big H. Because he has X, Y, it's easy. Look at this one is affected, so he has to have the small h. This one is affected, so he has to have the small h. This one is normal, he must have the capital H. This one is normal, he must have the capital H. Small h for him and a big H for him. So that's easy. The reason why we don't put something on the Y is the Y chromosome does not carry any alleles. So in a male, even if he has one, he is affected. Why? Because he doesn't have another X to counteract the effects of the small h there. Whereas a female, she could be normal, but she could be normal homozygous, or she could be normal carrier. That means she has that one, but she has a small one. Because of complete dominance, she won't show this color blindness or hemophilia, etc. So in this particular case, we need to know about her. But look at this one here, always in a pedigree diagram, look higher and look lower. So if you look at Sam there, she has to have the capital H because she's normal. But notice her son Jabu here. Jabu has the small h. Where did he get the small h from? He got it from her. Because he can't get the X from his father. If he got the X from the father, he would have had two X's. And he would be a female. So no good. So he got the small h from his mother. So that's how you figure out what is happening where. Okay, so that's what's going to happen there. Here's Sabi now. Again, she has a child, so she had to have the small h. But she was normal, so she had to have the capital H. So that's how we know whether a female is homozygous for the condition or heterozygous. And we can go right down there. Let's see what they want to know. Write down all the possible genotypes of the following individuals. Jubes, we've worked out already. H, capital H, H, small h there. All right? No, that should be H, small h, sorry. That should be H, small h. Jubes, remember, he is there. Jubes is hemophiliac, and hemophiliac is caused by the recessive one. And the next one is Sabi. She has to be heterozygous. We've shown that earlier. Why? And Yusti, let's go back to the diagram. Yusti is this one here. And it's normal, so that's straightforward as well. So therefore, we have that there. But we must also look at Yusti in terms of Romana, but because he's not there, oopsie, because he's not colored, he has to be that there. It can't be a depending story there. Okay? From the pedigree diagram above, state the relationship between gender and hemophilia. That's straightforward. Hemophilia occurs in males only. It is more prevalent in males. And why so? Because the females, they have two chances at the pie. In other words, they have two X chromosomes. So if the one X chromosome has the small h, there is another chance of the other one having the capital H, so they have that. The male, on the other hand, the minute he has the small h, he is hemophiliac already. Right? Illustrate a cross between Mo and Romana if Romana is a carrier of hemophilia. Mo, let's go back. Mo is a hemophiliac. So Mo is hemophiliac. Romana is a carrier. So Mo is X small h, X Y, whereas Romana is X big H, X small h. The gametes here will be X small h, Y, X capital H, X small h. And if we combine them, our Punnett square now, let's draw a Punnett square for that one. We have X, go back up, small h, Y, X capital H, X small h. X small h, X capital H, it will be X small h, X capital H, X small h, X small h, X capital H, Y, X small h, Y. Look at this combination. If the mother is a carrier and the father is hemophiliac, the chances are 50% of hemophilia and 50% normal. And that is 1:1 if we bring it down to its ratio. This is how we work out a pedigree diagram. Remember, we can also work out pedigree with other issues as well. If we look at the next one, they're talking about the pedigree diagram of albino inheritance. Now here, albinism is not sex-linked. So we use normal letters A and small a to do that. Also recessive gene, so therefore, this one is normal, normal, but we have an albino here. So if this albino came from these parents, it means both parents had to have a small a. This is how you work it out. So this child is small a, small a. To be albino, you have to have small a, small a. So in a diagram where there's no sex link, you simply go and do the recessive ones first. Now because this child has this small a's, two small a's, it could have got from there and from there. So both must have it. But they are normal. So they will have the capital A. That's how we determine whether normal ones are homozygous or heterozygous. We either look above or we look below. So we have that situation there. This normal one, we won't be able to say whether it's AA or AA because there's no further links here. This one is normal. Again, it could be AA or AA. Here, but now, this one here is normal and is having this one here. Has to have a small a there, and that one also has to have a small a because this child has one from there and one from there. This one here, we won't know whether it's AA or AA. I've done all that on the story there, so I don't have to go into the details there. And let's look at question 3.2.2 now. They're saying if individual Q is heterozygous, heterozygous would be AA there, right? Small a. And what are the chances of individual P and Q having a normal child, right? Now, P we know was albino. So that's the story there from our diagram. So all we're doing is a phenotype. If P, the phenotype will be albino, and here we have a carrier, normal, but it's a carrier. So the genotype obviously of the albino is small a, and the carrier is capital B, A, small a. When meiosis takes place, the A's are separated, the alleles are separated. And now if we have fertilization takes place, notice that no matter how we work, the format is still the same. That one and that one gives you small a, big A, and that one and the small a gives you a, small a, small a. And the same is going to be repeated with that guy, A, capital A, small a, small a. So you can see that again you're getting a 50% chance of albino and a 50% chance of normal. And this normal is just not homozygous but heterozygous normal. And this is how you work out the diagram in this case. Remember, this is also a pedigree diagram, but it's given differently. There you are given squares and circles, and it doesn't mean that when they talk about male and female all the time that it has to be sex linked. They can talk about deafness and albinism and males and females as long as they don't say that it's sex linked. But if it's hemophilia and color blindness, they don't even have to tell you that because you're supposed to know that hemophilia and color blindness are caused by sex-linked diseases. With that, have a small break. When we come back, we tackle the more difficult one of dihybrid. Welcome back, Mindsetters. Remember, up to this point, every problem, whether it was complete dominance, whether it was incomplete dominance, or it was codominance, or it was blood groups, or disorders, sex-linked disorders, we looked at one characteristic at a time. That is called monohybrid crossing. That means we're only looking at one characteristic at a time. However, when a baby is born, it's not only first only the eyes are formed and then the nose and so on and so on. When the baby is born with all of these things. And therefore, it has inherited all these characteristics already in the sperm, and not in the sperm, sorry, in the fertilized egg. So therefore, sometimes more characteristics are done at the same time. And that we call a dihybrid cross when we look at two characteristics at a time. A rooster with white feathers and a yellow beak. Can you see they're talking about two characteristics? They're looking at feathers and they're looking at the beak. Mated with a hen with black feathers and a red beak. So we're looking at feathers and beaks. Feathers are either white or black. Beaks are either yellow. Look at my homework that I'm doing. I'm writing this. You should be writing it on the end of your question. Beaks are yellow or red. The F1 generation consists of 12 chickens, all with white feathers, but 9 had yellow beaks and 3 had red beaks. Use the symbols F and F small f for feather color and B and small b for beak color. Now this is a dihybrid cross, so it's complicated. Remember, first master the monohybrid crosses before you come here. The fact that they said 9 had yellow beaks and 3 had red beaks. All had white feathers. That means white feathers will be capital W and black feathers will be small w. I would prefer rather to use capital B. There they told you there, sorry, I don't prefer, they're telling you, B and small b for beak color. And F there. So capital F for white and small f for black. And capital B for which color? Yellow, because 9 had yellow. And if you know your dihybrid cross, when we cross those things and we get 9:3:3:1, that means 9 would be of the dominant and the dominant. Dominant meaning dominant feather color, dominant beak color. So 9, that would be yellow beaks would be dominant, that means. And 3 had red beaks, that will be red beaks there. So name the recessive characteristic from the cross given above. What is recessive? Recessive means the one that does not show up if they're together. So in terms of feathers, it will be black, and in terms of beaks, it would be red. Right? Black feathers and red beaks given from our story above. Give the genotypes of the rooster and the hen. Which rooster and the hen? The one that were mated. And the genotype of that is obvious. The rooster had to be homozygous for the condition of the feather color and heterozygous for the beak color. Because if the beak color was homozygous, there's no chance in the offspring to have both red and yellow. But if we're looking at this offspring, we're finding red and yellow beaks. So that means this parent had to be heterozygous for it. And whereas the hen was homozygous for both conditions. Why? Because they had, the hen was black feathers and red beaked. And both these are recessive characteristic. And a recessive characteristic can only show itself when it is in the homozygous condition. FF small and small b for beaks. Give the possible gametes produced by the rooster. Now here I want to show you a trick. When you want to work out the gametes, the rooster's genotype was capital FF and big B small b. Capital F, big B, small b. In this dihybrid cross, the easiest way to work out the gametes, you do a small monohybrid cross. So you put capital F, capital B, small b. That will give you capital F, capital B, capital F, small b, capital F, again capital B, and capital F, small b. So these are your gametes that you're going to get. That's the easiest way to work out the gametes in a dihybrid cross. That's what we're getting, capital F, capital B, capital F, small b. Write down all the genotypes of the F1 generation. And that's our genotypes you're going to get there. And that. Illustrate the original cross with a Punnett square. Remember, first you're going to work out the gametes, and then you're going to work out the genotypes and so on. So the first one we've already worked out. It was FF capital B small b. So it will all be FB and F small b. All will be the same. The second one is even easier. It's small f, small b, small b. So all will be small f, small b. So if we put it together now, we've got capital F, capital B, capital F, capital B, capital F small b, capital F small b. That was from the rooster. From the hen, we're only getting small f small b, small f small b, small f small b, small f small b. So there's only two types that we can get from here. If we take this one, we can get one. Obviously, these two give you the same. So it's capital F. We can't put capital F now, capital B, because when they come together, the homologous chromosomes are together. So feather color will be dealing with feather color and beak size. So from there, you're going to get the F from there and the small f from there. They come together. And the capital B from there and the small b from there. And obviously, all these are going to be the same because all these are the same, and this one is the same. And this line is going to give you the same thing, capital F, small f, capital B, small b. This line and this line will be the same, capital F, small f, small b, small b. And obviously, all these will be the same, and all the b will be the same. Remember, in a different problem, if these things are different, then all these will be different as well. State Mendel's principle of independent assortment. Alleles pairs. Remember, alleles are in pairs. But when they separate independently during the formation of gametes. What does this mean? It means that, say for example, we have an allele for feather color and we have an allele for beak color. When these gametes separate during meiosis, the feather color does not affect the beak color and the beak color does not affect the feather color. Why? Because they are probably on different chromosomes, so they separate differently, independent assortment. Do not confuse this with a random assortment where in meiosis metaphase 1, we say that the chromosomes are lying randomly at the equator so that when they separate, there's no guarantee which sperm or egg will have which allele. Whereas here in independent assortment, we're saying that the different alleles for different characteristics segregate independently of each other, so the one does not affect the other. So this is why we get this setup in dihybrid cross. Remember, the difficult part in a dihybrid cross is to work out the gametes. And the easy way to do that is by using a small monohybrid Punnett square to work it out. All you're doing is you're taking the one allele from the one characteristic and you're combining it with another allele. Because what we are saying that in the sperm, you can't get FF because that means the sperm got an extra chromosome. So therefore, that is not allowed. So it can only have one F and it must have one of the B's. Can have a capital B, that's one type of sperm. Another type of sperm has a capital F and a small b. These are the possibilities. Obviously, that holds true in an egg as well. If that was an egg, we're getting that type of situation there. In our example, all the eggs in the hen would have had small f, small b. This is in the rooster. This is what we can get. This is what we showed here. I've just shown it in eggs as well so that you understand that it can be in an egg or a sperm. But in this particular case, the rooster, these are the possibilities. Not this one here, remember. This is not allowed because if that is allowed, that means it has an extra chromosome. Because every gamete only has one of that particular allele. Why? Because during anaphase of meiosis, the homologous chromosomes separate. So that means the sperm or the egg has only half the chromosome number. When the mother and the father, the sperm and the egg come together, then now the resultant zygote has both again. So that is what we are showing. Guys, I'm hoping that this helps you to understand dihybrid crossing and all the other examples we have done. and I'm hoping that this enhances your knowledge on genetics in general. With that, I wish you well. Enjoy the rest of your day.